Good morning, dear students. Today we are going to observe Rhizobium leguminosarum in the root nodule of MIMOSA pudica. And this practical is for second MSc Zoology students. Okay, let us start the practical. Before that, we need to have some basic knowledge regarding what is root nodule and what is the nature of that bacterium that we are going to see today, that is Rhizobium leguminosarum. First, we are starting with the root nodule. What is root nodule? It is a site that we can see in some roots of some leguminous plants. Here in this picture we can see here, and these are the site of symbiotic association between a type of bacteria that we know that here it is Rhizobium leguminosarum, and it make an association with this plant, and that association is called symbiosis. Then what is symbiosis? Symbiosis means it is a type of association between two species, and in that association sometimes both the partners may benefited, or sometimes only one is benefited and another get harmed. That may vary, but in this case both the partners are benefiting. Then what benefit they are obtaining out of this association? That is what we need to understand first. In the case of this plant, it needs nitrogen for its growth, and it cannot fix the atmospheric nitrogen by its own. For that purpose, this bacteria that is present in the this symbiotic association, it helps this plant to fix the atmospheric gas to ammonia, and later this ammonia get assimilated in the plant in the form of amino acids or nucleotides, or in the form of some vitamins or hormones that all are useful for this plant. In that way, that association is beneficial to this plant. Then what benefit this bacterium is getting out of this association? In the plant, because of its photosynthesis, a carbohydrate called sucrose is forming, and as a result of its breakdown, another product called malate is forming, and this malate is the immediate carbon source for this bacteria. In that way, this bacteria also benefited out of this association. Okay. Next, I am going to explain about how this nodule is forming. The roots of this leguminous plants, I will draw here. Suppose this is the roots of the leguminous plants. They produce a secondary metabolite called flavonoids. And these flavonoids attract the bacteria, Rhizobium bacteria, that present in the soil in the vicinity of these roots. This is the bacterium, and as this bacterium are attracted by these flavonoids, some genes that present in the bacterium, they are called the node genes. They are called the node genes. They get activated, and under the influence of these node genes, the root hairs of this root start to curl. Why? What is the purpose of this curling? To entrap enough number of this type of bacteria that is present in the vicinity of this root hairs. And when enough number of bacteria is entrapped, then it forms a colony there. And as we examine this root hairs closely, then we can see that I will show here. Suppose this is a part of root hair. A tube called infection tube is developed inside this root hairs, and through this tube, infection tube, that Rhizobium bacterium gain an entrance inside the root. As it enters the root, it pierces the epidermis of the root and reaches the cortex. And in the cortex, it gets wrapped by a membrane called symbioso membrane. What is the name? Its name is symbioso membrane. After reaching the cortex of the root, and as it is enclosed in that membrane, then it became active and start to fix the atmospheric nitrogen for this plant. And how that process initiated? First, it prompted that root to form some projections. That projections, that swellings are called nodules. That nodules start to appear in the root, and in that nodules, this bacteria start to fix this nitrogen. Okay, and here in this image, I am showing the appearance of this Rhizobium leguminosarum bacteria. It is somewhat rod shaped. In some case, it is appears oval. Anyway, we can go and see the this bacteria in the root nodule today. And for that practical, the materials required are root nodules, 2 slides, needles, methylene blue stain, cover slip, beaker, and microscope. Okay, let's start the practice. First, this is the root nodule. We can clearly see here. These are the root nodules. Now we have placed 1 or 2 root nodules in a slide, and we are just going to smash it. We can smash it in 2 ways. Either using the needle we can smash it, or we can press it between 2 slides. In that way also we can smash. Okay, then next we need to remove the waste part of the root nodule from that slide. Next we are going to make a smear. For that we need to keep the slide in this way and place the other one over it in a 40-degree angle and make a single swipe. Then only we will get a nice smear. Okay, now next we are going to heat it to fix the bacterial cell in that slide, just passing over the flame. Okay, next we are going to add the methylene blue stain over the slide to stain the bacterial cells. We applied the stain. Then for spreading the stain, either we can tilt the slide or to shake it to make it to spread all region of the smear. Okay, then we kept it for drying for some time. Okay, then next we are going to wash out the excess stain from the slide by dipping the slide in the water that taken in the beaker. Okay, so just dip it and take out. Okay, then again we need to dry it. Keep it for dry. After drying it properly, then only it is in ideal condition to observe under the microscope. So it is kept for drying for some 2 or 3 minutes. Then we are going to put a cover slip over that bacterial cells and observing under the low power of microscope. Under the microscope, we can see the Rhizobium leguminosarum in this form. Irregularly shaped cells. Some cells are in oval form, some cells are spherical. Okay, for the examination, you will be asked to draw a picture of this bacterium also. Okay, this we can finish today's practical. Thank you.
I'm an icon, man. My girl about a 10. I should fuck her in the Benz.